Welcome to Bacteriophiles Micro Edition. Get ready to hear a recent exciting story about microbes with your host, Jesse Noir. About 10 years ago, scientists discovered a virus in an amoeba. They called it mimivirus. It was the biggest virus they had ever found. They actually mistook it for a parasitic bacterium. It was that big inside the amoeba. You could see it under a light microscope, like bacteria, and it's actually larger, its genome is larger than some actual bacteria, parasitic ones. Then a couple years ago, another virus called megavirus was found, which is even bigger than mimivirus. It's also found in amoeba. This virus has its own genes that almost all other viruses borrow from the host to make its proteins. So instead of hijacking a lot more stuff from the host, it makes it itself. And this megavirus actually has its own virus called a virophage named Sputnik that depends on the megavirus for its own replication. Now, the same scientists that discovered megavirus, publishing in Science this past July, have discovered an even larger kind of virus called Pandora virus. They were looking for large viruses in samples of sediment in the ocean, samples that can lyse amoebae pretty well. From these samples, they chose ones that had amoebae with particles inside them that were large and replicate, and also that were insensitive to antibiotics, because if it's sensitive to antibiotics, it's probably a bacterium. So they found two in particular. The first was from sediment about 10 meters down at the mouth of the Tunquén River in Chile, and the second from the other side of the world, from pond mud in Melbourne, Australia. Both of these were oval-shaped and about half the size of E. coli. They were that big. So the scientists observed the whole life cycle in the amoebae. It took about 10 to 15 hours. First, these particles were taken up by phagocytosis, so the amoebae engulfed them and consumed them. And then the particles emptied out their contents into the cytoplasm of the amoeba, similar to mimiviruses. Then after a while, the host nucleolus, the structure inside the nucleus, disappears, and the nucleus changes shape, and then it disappears too. And the cells round up and look pretty sick, and then new virus particles start to appear where the nucleus was. And finally, the cell lyses, breaks open, and releases about 100 more particles to go infect other amoebae. So what's interesting about that is that the megavirus and mimivirus pretty much stay in the cytoplasm rather than going into the nucleus, and they make their own replication cycle in the cytoplasm. But this one appears to require the nucleus for its replication. So they sequenced the genome of the Pandora virus, and found that in terms of number of bases, it's about half the size of E. coli's too. This is even bigger than some eukaryotes, some parasites. That's just crazy. The two viruses from the different parts of the world were pretty similar to each other, except for 4 regions that were unique to the one from Chile. And about 400 of the 2,500 potential genes match previously known genes that we've found before, but the rest of them don't match anything. Of the ones that do match known proteins, there were about 200 that weren't kind of copies of each other. And of these 200, about half of them matched eukaryotic genes, and the rest were split between bacteria and viruses. So overall, it doesn't seem to be closely related to any other known thing that we've discovered. And because of this, the researchers actually worried that the virus might be using a unique genetic code. So almost all life uses exactly the same genetic code, converting the DNA to proteins, but some things do it a little differently, and it's always possible that there could be a different code. So they actually thought of this and tested it by analyzing the proteins that were predicted to be coded from the standard code, and they found that it does use the standard one, so it just has a lot of weird genes. And not surprisingly, it is lacking in most of the expected components for cellular organisms that cells would need to survive for translation or energy generation. but it does have some genes that are associated with the other large viruses and unusual to most other viruses. But unlike the other large viruses, as I said, it seems dependent on the host nuclear proteins. So this is pretty amazing. It can be relevant to humans because amoebae are pretty similar to macrophages or white blood cells. They go around and eat bacteria and viruses. So it's possible that viruses that infect amoebae might someday come to infect humans and be able to get around our immune system defenses. It also could be useful because it has a lot of unknown genes, and some of them could do useful things, either to produce better medical treatments or other kinds of stuff. The authors speculate that this virus and others like it could represent a fourth branch of the tree of life, in addition to eukaryotes, bacteria, and archaea. This is based on the sequence of the DNA polymerase that copies DNA. When you compare the different sequences from these viruses and the other forms of life, it seems to group together on a fourth branch by itself. The authors seem to think of this kind of virus and maybe other viruses as another kind of life form, whereas some people disagree that viruses should be considered alive because they can't replicate on their own. The authors distinguish them from what they call cellular organisms, so I guess the implication is that these viruses are non-cellular organisms, or acellular. Rather than being their own organisms, maybe it's more likely that the viruses could have come from free-living organisms, other kinds, and just lost the ability to live on their own, and just depend on cells. It seems weird that there would be a whole fourth branch that is represented only by viruses now. There aren't any free-living members of it, but there's always a possibility that we just haven't found them yet. One thing that this paper demonstrates is how important it is to be open to possibilities, and you won't find something if you're not open to it or looking for it. So it seems that people might have seen this virus even 13 years ago, but they just assumed it was a bacterium. And I was impressed at how they considered the possibility that the virus might be using a different kind of genetic code and tested for it. So it's pretty good science. Just shows how much remains unexplored in our world. The microbial world is still so mysterious.
That's all for today's Bacteriophiles Micro Edition. Send comments or questions to bacteriophiles@gmail.com or comment at bacteriophiles.blogspot.com or at microbeworld.org/bacteriophiles. The music is from Nevio's PodSafe Music Alley at music.nevio.com. Thanks for listening.